Welcome to the class, everyone. I am Dr. Shailendra Singh from Department of Biotechnology, and the topic today we are going to discuss is secondary metabolite production in plant tissue culture. So, as we talk about secondary metabolites are synthesized by the plants, and we are producing them with the help of suspension culture of many species. These plant species accumulate certain biochemicals which we refer to as secondary metabolites. These are present in very high concentrations in some plants. They are mostly alkaloids, steroids, terpenoids, flavonoids, and some related compounds and quinones, which are valuable as drugs, flavors, fragrances, colors, etc. The yield of chemicals sometimes exceeds 20% on dry weight basis in some plants, and in some cases it may be low. There are some specific cases of commercial production of these biochemicals from plant cell culture. For example, chiconin, that is an important drug and precious natural dye for silk and cosmetic. It has been produced by the cell cultures of Lithospermum erythrozoon. The chiconin yield from cell culture is very good. It is around 20% on dry weight basis. Whereas in intact plant, it is around 1.4%. So that's how we can scale up the commercial production of these secondary metabolites with the help of plant tissue culture. Similarly, berberine and Taxol, these are also being produced from cell cultures. In addition, ginseng biomass is also being produced in vitro. So today we are going to start with the production of such secondary metabolites from cell culture. But first we will discuss about the secondary metabolites and their use. So these higher plants synthesize a wide variety of low molecular weight compounds. These are low molecular weight compounds, and they are produced by the plant in addition to the essential primary metabolites. Primary metabolites are essential as these are needed for the plant cell and growth. And secondary metabolites are mostly used by the plant in their pollination and in their defense mechanism. These secondary metabolites offer protection against pests as they act as attractants also, and as these plants does not have, they are basically plants on hormones, as insects have pheromones. So plants have these fragrances with the help of these secondary metabolites. Why chemically warfare to protect plants from the attacks by predators, pathogens, or competitors, because as they are providing protection to the plant against predators, pathogens, or competitors. These secondary metabolites attract pollinators like insects, honeybees, and seed dispersal agents also. These are giving plant protection against abiotic stress also. Abiotic stress are the stress conditions produced due to several climatic or environmental conditions, like it may be due to excess or deficit of water, it may be high or low temperature, it may be presence of some salt or acid factors in the soil. Likewise, these secondary metabolites are also used as medicines, and they are also being used as industrial additives. So these secondary metabolites have immense importance in several walks of life. That's why we are interested in the production of secondary metabolites. If we come to the classification of secondary metabolites, chemically their structure is different, so they can be classified on the basis of their chemical structure. on the basis of their chemical composition, on the basis of their solubility in various solvents, or by the pathways by which they are synthesized in the plants. So there can be different classification for these secondary metabolites. A simple classification of secondary metabolites include three main groups. What are these main groups? First, the terpenes. Second are the phenolics. And third are the nitrogen containing compounds. So terpenes are made from malonic acid, which is composed almost entirely of carbon and hydrogen. Phenolics are made from simple sugars containing benzene ring, hydrogen and oxygen, whereas nitrogen containing compounds, they are extremely diverse and they may also contain sulfur. So first category we are going to talk about are the alkaloids. You must have heard this term. Most are derived from a few common amino acids like tyrosine, tryptophan, ornithine, arginine and lysine. These compounds have a ring structure and a nitrogen residue. Indole alkaloids is the largest group in this family, which is derived from tryptophan. And these alkaloids are widely used as medicine. What are the characteristics of these alkaloids? These alkaloids are bitter in taste. They are derived from amino acids. The amino acids that are most often serve as alkaloidal precursors. Alkaloidal precursors means the substance from where alkaloids are being synthesized. प्लांट्स में वो केमिकल जिनसे एल्कलॉइड का सिंथेसिस होता है, एल्कलॉइड्स बनते हैं, उन्हें हम एल्कलॉइडल प्रीकर्सर्स क्या है? अमाइनो एसिड्स, क्योंकि एल्कलॉइड्स किससे बन रहे हैं? अमाइनो एसिड से. कौन कौन से अमाइनो एसिड्स हैं? दीज आर फिनाइलएलेनिन, टायरोसिन, ट्रिप्टोफैन, हिस्टिडीन, एंथ्रेलिनिक एसिड, लाइसिन and ornithine. So these are the precursors for alkaloids. Alkaloids form double salts with compounds of mercury, gold, platinum and other heavy metals. These salts are obtained as precipitate, which are microcrystals, so they can be crystallized also. जब भी आपको एल्कलॉइड्स का एक्सट्रैक्शन करना है, आप उनको प्रेसिपिटेट कर सकते हैं और क्रिस्टलाइजेशन भी किया जा सकता है. These are insoluble or sparingly soluble in water, but the salts formed on reaction with acids are usually freely soluble. Most are crystalline solids, although a few are amorphous also. Free alkaloids are usually soluble in polar solvents like ether and chloroform. So free alkaloids. Salts are soluble in water. Salts of alkaloids are soluble in water. But if you have pure alkaloids, then you can dissolve them in polar solvents, either in ether or chloroform. Some alkaloids are liquid because of lacking of oxygen in their molecules. And liquid alkaloids examples are conine, nicotine and spartanine. What are the sources and occurrence of alkaloids? आप alkaloids अगर plant kingdom में search करेंगे, where you will find the alkaloids. So they are present among different families, mainly leguminaceae, legumes family, जहाँ से आपको pulses मिलती हैं. Papaveracea, opium poppy is an example. Ranunculaceae, rubiaceae, solanaceae. Again, solanaceae is a very important family in our daily life. We have potato, tomato, and brinjal from this family, and barbadeaceae. These are outstanding alkaloid yielding plants. What is the use of alkaloids in plants? These are poisonous agents which protect plants against insects and herbivores. So it is very, very good. They have a very important role in the survival of the plant. and product of detoxification reaction representing a metabolic locking up of compounds otherwise harmful to the plant. That means these alkaloids, these are the end product of detoxification reaction. So they are not being ended up in a toxic product. Otherwise, that will be a harmful effect on the plant. But in other end, they are providing a protective role to the plant by the accumulation of these alkaloids. They are regulatory growth factors. They have reserve substance capable of supplying nitrogen or other things, other elements which are necessary to the plant growth. In case of stress condition, they can be substituted as plants nutrition factors. Second category are the terpenes. After alkaloids, second is terpenes. These are natural products that are structurally related to isoprene. What is isoprene? If you see this structure, this is just for your reference. If you understand what is the isoprene structure, it has how many carbons? It has 1, 2, 3, 4, and this 5. So it has 5 carbon. So this is the basic unit of terpenes. So isoprene, if one isoprene is there in a terpene, that is monoterpene, it will have 5 carbons. If I say diterpene, then there will be 2 isoprene units and there will be 10 carbons. Likewise, triterpene, there will be 3 isoprene units and there will be 15 carbons, so on. So in terpenes, carbons will be in the multiple of 5, depending upon the number of isoprene units they contain. The number of carbon atoms is a multiple of 5, 3, 5, 10, 15, 20, likewise. Each group of 5 carbon is an isoprene subunit. They can be saturated or unsaturated. Saturated means there will not be any double bond or triple bond. Unsaturated means there may be presence of double bond or triple bonds. Many contain oxygen atoms as well. Now the third category are the phenolics. They are derived from aromatic amino acids. Which are the aromatic amino acids? Aromatic amino acids are phenylalanine, tyrosine, and tryptophan. So phenolics are made up from phenylalanine, tyrosine, or tryptophan. For the production, for the synthesis of phenolics, the presence of these aromatic amino acid is necessary in plant system. All contain structures derived from phenols. What is the basic structure of phenol? Phenol is a benzene ring having OH group. Some examples, one is coumarins. Coumarins are antimicrobial agents, feeding deterrents. Feeding deterrents means like your crop is having grains, so birds can come, visit these plants and they can feed on the grains. But due to the presence of phenolics, they may be bitter in taste, they may have strong aroma, so birds will not come and feed on the grains. Feeding deterrents are good for the plant and germination inhibitors also. Second example of phenolics is lignin. It is abundant in secondary cell wall. It gives rigid and resistance to extraction or many degradation reagents. Then there are anthocyanins, flavones, flavonols. So phenols are present in every plant. They attract pollinators to the plant and even impact how these plants act with one another. That means they are helping in plant to plant communication also. Examples of glycosides are glucosinolates. They are found primarily in the mustard family to give the pungent taste. So sometimes the pungency varies in your mustard oil. There is a different degree of pungency in different mustard oil, that is due to the amount of glucosinolates. If the glucosinolates amount is high, then pungency would be very, very high. So there are different type of linkage which are present between glycon and aglycon. So what is glycon? In glycoside, the carbohydrate part is known as glycon and the non-carbohydrate part of the glycoside is known as aglycon. So the bond with the carbohydrate part is known as glycosidic bond. So it may be C linkage, it may be O linkage, it may be N linkage, or it may be S linkage. So what are the sources and effects of major secondary metabolites? If you see these fruits, these are so beautiful, colorful fruits, very abundant in this season. So these are capsicums, oranges, carrots. So what is common there? You can see that they are mostly having the similar color, red-orange color, that is due to the presence of carotenoids. Carotenoids are organic pigments occurring in plants and are mostly found in red, orange, and yellow fruits and vegetables. What is good thing about consuming these carotenoid rich fruits and vegetables? They have antioxidative effect and prevents cancer. Why antioxidants are good? Because antioxidants fights free radicals. Free radicals present everywhere. They enter our body. They are present in pollutants, enter our body, and they damage our cells. So if there is no antioxidant in our body, these free radicals will keep damaging our cells. That's why they accelerate aging effect in humans. So if you are consuming enough antioxidants, these antioxidants in your body can fight with free radicals and your cells will not be damaged. Your cells will remain healthy. They will not have effect of the free radicals and their amount can prevent cancer also. They boost your immune system and reduce the risk of heart attacks. So consume a lot of orange, yellow, red fruits and vegetables to have more amount of carotenoids. Now the second category is phytosterols. So what you see here, these are different type of seeds. These are pumpkin seeds, flax seeds. So these phytosterols are found in sunflower seeds, sesame seeds, nuts, and soybeans. They protect against colon cancer and low cholesterol levels. So you should consume nuts and seeds for getting phytosterol amounts in your body. Next are the saponins. So what you see here, these are different type of pulses. Green gram, black gram, rajma, lobia is there. So these are flavor additives found in legumes and spinach. They also boost the immune system, lower the cholesterol level in blood, and reduce the risk of intestinal cancer. Next example are the flavonoids. These are very, very good for our health. As you can see here, different type of fruits, cherries and berries. So these are the organic pigments, gives plant red, violet or blue color. Inhibit the growth of bacteria and virus, protect the cells against the damage of free radicals. That means they also have a lot of antioxidants. So they also have antioxidative quality and they also influence blood coagulation. Next category examples are sulfides. Sulfides, as the name indicates, what you see here, you see onions and garlics. So they contain sulfur, belongs to the lily family. Onions, leeks, asparagus, garlics inhibit the growth of bacteria, lower cholesterol levels, and they also have preventive effect against cancer. Next example are curcumin. This is very popular ingredient. In this pandemic times, everyone is advising to you to have a lot of curcumin in your diet. So this secondary metabolite is extracted for turmeric. Botanical name of turmeric is Curcuma longa. So for curcuma, the curcumin name has been given to this secondary metabolite. It has coloring and medicinal use, and if you see the medicinal use of this curcumin, it is very effective in wound healing. It has anti-ulcer property, it has anti-inflammatory property, it has antimicrobial, antiviral property. It is hepatoprotective in nature, it has antioxidant property, and plus point advantage is that it does not have any toxicity. So you can consume curcumin in your daily diet for having so many advantage of this curcumin. Next example are these flavors. So what are these? These are the cocoa seeds, these are the vanilla beans. So normally these are different flavors which you use in your daily life. It may be obtained from cocoa seeds, vanilla beans, strawberries, cinnamon, cardamom, cloves, pepper, or areca nut. So why they have these flavors? Due to the presence of different secondary metabolites in their fruits or flowers. Different parts of the plant have different secondary metabolites. Now come the part where we are discussing how can we produce these secondary metabolites in plant tissue culture in the lab or in the commercial scale by the help of bioreactors. So for this, what we need here, very important part is the choice of explant. Why the choice of explant? As we know that these secondary metabolites are very specific to the particular organ of the plant. For example, if you talk about the mentha oil is produced by the mint leaves. Mint leaves have the glands which produce mentha oil. So if you want to do the production of that oil in cell culture, you have to take leaf as explant. So in case of secondary metabolic production in plant tissue culture, morphogenic differentiation of the explant is very important. You have to take a specific explant for specific secondary metabolite production. Any part of the plant can be used, but for the callus or tissue induction, but here prokaryote explants, these prokaryote explants are sterilized prior to culture by using ethanols. So what is the procedure of disinfection of explant? You will take the specific explant from the mother plant, wash it with the liquid detergent, then you can treat it with either with antibiotic or you can treat that explant either with antibiotic or with a fungicide, and then it is treated with the disinfective agent in laminar airflow cabin. A disinfection agent may be of your choice. You can use either ethanol, sodium hypochlorite, calcium hypochlorite, or mercury chloride also. Then comes the nutrient culture media. For the production of secondary metabolites, which medium you will use? So first you have to induce callus from an explant, and then the callus is cultivated in the suspension culture. So you can use the same media for both process, for callus induction and for cell suspension culture. or you can use different media for different actions. For callus induction, a callus culture is usually sustained on gel medium. Callus induction medium consists of a gas, solidified medium we will take, and a mixture of macronutrients and micronutrients for the given cell type. There are several types of basal salt mixture which are used in plant tissue culture, but most notably modified Murashige and Skoog medium, which we popularly know as MS medium, White's medium, woody plant medium. Woody plant medium is known as WPM. Vitamins are also provided to enhance growth, such as Gambox B5 vitamins. For plant cells, mainly the components which we require are nitrogen, phosphorus, and potassium. NPK are especially important, so they should be supplied in the nutrient medium in sufficient amount so that the callus induction takes place in no matter of time. That means it develops easily and rapidly. Then comes the suspension culture. Growth rate of suspension cultured cell is generally faster than that of solid culture. Here we are taking a liquid medium that is devoid of agar powder. The former is more desirable, particularly in the production of useful metabolites on a large scale. So in commercial production, lesser the time taken for the production of secondary metabolites, that is the better. When we are doing it on commercial level, then we will be doing it in either fermenter or most efficiently known as bioreactors. So bioreactors, either it may be a stirred jar fermenter, or we can use it for basically this type of fermenter is used for microbial cultivation at a very low agitation speed. If you can see the diagram here, it is a basic fermenter or basic bioreactor. It is made up of, on commercial level, the body is made up of stainless steel and it has this double layer outer covering. Why this is double layer? Because it acts as a cooling jacket or sometimes as heating jacket also. If you want to maintain the temperature of this inner space, so what you will do? Accordingly, if the temperature is high, then you can run cold water in this tube. The temperature will drop down. If the temperature is down and you want to increase it, then you can circulate hot water or steam in this wall or in this tube. Then it will help you in increasing the temperature. It is having the medium plus cells are there. For the efficient mixing of nutrient medium and these plant cells, here a shaft is fitted with these blades which is rotated with the help of a motor at a gentle speed, and when these blades moves inside this solution, then we have uniform mixing of cells with the nutrient medium. This is a feed port with the help of which you can inoculate the nutrients when the incubation is going on. These are the baffles. Baffles are the metallic strips which are readily attached to the wall of this bioreactor, and they prevent the formation of vortex inside this vessel. So this is the basic structure of this bioreactor, having agitator, baffles, this feeding port, and this is the motor which rotates these agitators. Sometimes hairy root culture also helps in the production of secondary metabolites. What is hairy root culture? It is a culture produced after the infection of explant and cultures by the gram-negative soil bacteria, Agrobacterium rhizogenes. So rhizogenes strain of Agrobacterium, this species causes hairy root diseases. So after some modification, this bacterium allows to infect the particular plant, and then from that particular explant, root production will be enhanced. This process takes advantage of the naturally occurring hairy root disease in the dicotyledons. Agrobacterium recognize some signal molecules which are exuded by wounded plant cells, and then it becomes attached to it. Agrobacterium, as you know, is also known as natural genetic engineer. Why this is known as natural genetic engineer? Because this species, Agrobacterium rhizogenes, it contains the RI plasmid, that is root inducing plasmid. Wherever it will infect, the root production will be enhanced. So this bacteria genetically transferred part of this plasmid, which is known as tDNA, that is transfer DNA to the plant genome. The plant cell to proliferate by increase the rate of cell division, because this plasmid also has some sequences which are responsible for the production of hormones. Which hormones? Mainly cytokinins and auxins. So as cytokinins and auxins would be there, it will help in the cell division. And when cell division will take place, so it will help in the growth. So produce the hairy roots. And it also has signals for the opine production. Opines are very, very specific compounds which are unusual amino acid, namely octopine and nopalene. Sometimes agropine is there, and they are present in this agrobacteria, and they serve as nutrition source also because they have carbon and nitrogen. How you do the commercial production of secondary metabolites? Higher plants contain a variety of metabolic products which are useful as medicines, food additives, perfumes, etc. However, non-compromising reduction in plant sources, increase in labor cost and other problems in order to obtain these high value added substances from natural plants have forced to develop an alternative approach towards the use of plant cell culture for production of these metabolic products. What is the help of plant tissue culture? Most importantly, you are not damaging the plant sources. You are helping in the conservation of natural plant sources which are giving us such beneficial products. Number two, the production of these secondary metabolites will not be affected by the environmental conditions, because here the conditions are controlled in plant tissue culture. So your product will be of high quality, it will be of uniform type. So some pharmaceutically important chemicals, that is the last part we are dealing about. Some examples. So one is morphine alkaloids. Morphine alkaloids, for example, one is codeine. Codeine is an analgesic and it is a cough suppressing drug. It is obtained from Papaver somniferum, that is a poppy plant. So that is the flower of poppy. Also, morphine can be converted into codeine. Many workers have tried to produce codeine by undifferentiated cells of these plants, that is the callus, undifferentiated cells which are present in the callus. Example 2 is berberine. It is an isoquinoline alkaloid which is produced and distributed in roots of Coptis japonica and cortex of Philodendron amurans. Berberine is chloride used in treatment of intestinal disorders. Next example are tropane alkaloids like scopolamine and hyoscyamine. They are used commercially as anesthetic and antispasmodic drugs, occur in the leaves of members of the family Solanaceae, including dubiosa, datura, scopolia, and hyoscyamus. Then anti-tumor compounds. We know that tumor is a very, very important topic, or we can say it is a disease for which scientists have been researching to get new medicines, so which can protect our cells against the tumor cells. So these anti-tumor compounds are present in some plants. For example, you might have seen this beautiful flower. It is of Catharanthus roseus, also known as Vinca rosea, and in Hindi, common local name is Sadabahar. So it has very good anti-tumor compounds which are named as vinblastine or vincristine. These treat leukemias, Hodgkin's disease, and also have anti-tumor activity. Another example is ginseng. You might have heard this name. Roots of Panax ginseng, widely used as tonic. These are effective for genetic disorders, diabetes, and weak circulation. These roots of Panax ginseng contain saponins and sapogenins. Then next example, application of these secondary metabolites, they are used as food coloring agents. They have beautiful colors. You might have seen this. This is the most expensive spice globally, that is saffron. It is made from the stamens of Crocus sativus and used as flavoring agent and colorant also. Plants contain crocin, that is yellow pigment, safranal, that is fragrance, and picocrocin, that is bitter substance present in the saffron. So these are the three secondary metabolite which gives saffron its characteristic flavor, color, and taste. It is used in baked goods, soaps, cheese, and it is having medicinal value for different stomach ailments also. Next example is safflower yellow. This is a yellow pigment obtained from the floret of Cathamus tinctorius. Major pigment is carthamine. Carthamine is a quinoid form of isocarthamine used in baked goods and beverages. These are the natural colors. Chewing gums, chicle gum. Chicle is the raw material which is obtained from the latex of Acarus sapota. Chicle contains 60% resin and 15% of rubber. So this is natural chewing gum source. Resin consists of lutein-alpha amyrin and beta amyrin. Rubber consists of cis and trans 1,4-polyisoprene. And these are synthesized through mevalonic acid pathway again. And next example is Taxol. Taxol is a diterpene amide. It is extracted from the bark of wild-grown Taxus brevifolia, and it treats breast and lung cancer. It is known as Taxus baccata, which is known as Pacific yew tree. This tree takes years to develop, many years to mature, and if we cut down the whole tree for the treatment of cancer disease, that would not be sufficient for the entire world population. So it is best to develop this Taxol in cell suspension culture so that we can treat the global population who are suffering from breast cancer and lung cancer. This Taxol is also have very immense anti-tumor activity. So thank you so much for listening so carefully. If you have any question, you can ask me. The secondary metabolites are very, very important, not only for plants, but for us also. We have been using them in our different walks of life. We are using them as medicines in pharmaceutical companies. We are using them as flavors, aromas, fragrances in cosmetic industry and different walks of life. So it is better not to devastate the natural population of the plants, but to find an alternative way, that is by producing them in lab condition with the help of plant cell culture. That's how we will be getting the natural products, plant products. We are not having synthesized products, and they will be good for our health also, and we are not damaging the plant population, which are providing us with such beautiful and natural colors and aromatic compounds. Thank you so much. That was all from my side for the production of secondary metabolites in plant cell culture. What are the secondary metabolites? Different examples, why they are produced in cell culture, and how they can be used. Thank you so much. Question time now. Abin is here, Amos, Om, Avang, Cherison, Charurima, Clinton. I think it's your first class, Clinton. Welcome to the class. I just noticed that you are here. Dipesh, Hamyo, Divya, Dorji, Gracie, Gumchi, Ika, Avaka, Sabkas, where have you been? You are not regularly attending the classes. Why so? Kindly give me a reason. Kangmei, Jibesh. Jibesh, same with you. You are very inconsistent in the classes. Please tell me the reason. Yoshar, Laksharaj,